I think it's still on them.
So shall I wait a couple of minutes or start?
Yeah, maybe start there and just, they're all...
Right, first I'd like to thank you for the invitation to come speak here. It's my first visit to Mumbai. I'm enjoying myself very much. I'm going to tell you a little bit about intracellular transport by myosins, but I really want to add a little question mark to that, because by the end of the talk, I'll come around to the question, the point that Peter Hollenbeck made that he said might be crazy, and I would like to argue that he's not crazy. At the end about in this point about myosins and about what they really do in cells. So I'm very much an outsider here, not only on the basis of myosins, but on the basis of my career trajectory. So I've been talking to some students this morning about changing fields, but to give you an example of where I've been, in graduate school I cloned the genome of mouse cytomegalovirus and worked on latency. That made me interested, working on the mouse made me interested in mouse genetics. So as a virologist, I did some retrovirology first in the laboratory of Nancy Jenkins and Neil Copeland. Then the project I really wanted to work on opened up, and I studied the insertional mutagenesis of a very classical mouse coat color gene that I'll start on here talking about. And back in the late 80s, you needed something like this insertional mutagenesis to tag a gene so that you could clone it. I realize this seems very ancient to you, but that's the way it was back then. And I basically started learning cell biology as an assistant professor at UT Southwestern. That was my first faculty position where I was next door to Scott Brady. So it's been fun seeing Scott again. So I'll talk about myosins, but first I want to make a point. This goes back to what Peter Hollenbeck was talking about, and I've been rewriting my talk frantically so that I don't cover the same things, but I do want to cover the same theme. For example, we have this thing where we call some myosins conventional and some are unconventional. Well, I'll concentrate in this talk on the myosin 5 family, first because it's what I know best. Secondly, it's the family for which we have the best tools and the most knowledge. There are 3 members of it, myosin 5A, myosin 5B. Both of these are found in neurons in the brain. And I'll ignore myosin 5C, which is primarily epithelial. 5A interacts with everything. It's like a Swiss Army knife in the cell, and I'll give you a hypothesis as to why that may be true. Myosin 5B is much more limited in its distribution. It's very highly concentrated in the hippocampus and dentate gyrus, which made it easy to study long-term potentiation, its role in that. Then we have the myosin 2s, and this includes, of course, muscle myosin, which we understand so well. These were discovered first, and that's really the only reason. There's no biological reason to call these myosins conventional. Even though we all do it in grant applications and papers, there's literally no reason to call them conventional. And I'll show you why. Because if people have done evolutionary analyses, the best one I think is by Cavalier-Smith, where they looked at domain gain and loss among these myosins. So you can construct trees by looking at homology between these conserved head domains, but if you look at the domain gain and loss, it appears that the first eukaryote had 3 myosins, none of which were conventional myosin. Okay, and it included a type 5 myosin. So biologically, if we think biologically and try to put all that history, you have to remember we understand a lot about muscle myosin because you can take a chicken breast and homogenize it and make a lot of pure protein from it. But which are really more conventional? So here's the nice figure from that Cavalier-Smith paper. But basically they show this domain gain and loss. Here are your myosin 2s, very late in evolution. They required an extension of the alpha helical coil domain. That was the key event there. So they're very, very specialized, even though we as organisms have a huge amount of them. It's not, they're only conventional in terms of their abundance in the body, not in their function. But if you go back and basically the tree they constructed, they had 3 myosins, one of which was a myosin 5. So I got into this business in this mouse genetics lab I worked in, Copeland and Jenkins. We were studying the dilute mouse mutant. And I don't know if you know much about the history of inbred mice, but there were a group of mouse fanciers at the turn of the 20th century that bred all these mice with pretty coat colors and had all these interesting behaviors, such as waltzing. They had mice that did nothing but turn in circles their entire lives. And everyone thought they were interesting, and they called them waltzing mice. Well, it turns out that they're deaf, and so they've been a huge resource for studying deafness. The coat color mice are also interesting for another reason, because there's a huge connection between coat color phenotypes and neurological phenotypes, since melanocytes come from the neural crest. But anyway, so this mouse mutant was so old that it had a single letter gene symbol, of course D, and I just can't begin to convey my disappointment that it's now been changed to Myo5A, which is just not descriptive. I guess maybe I should have been a fly geneticist, because I like these descriptive names. And for example, it's been fixed in several of the common inbred strain. It's actually what the D stands for in the strain DBA. This actually stands for dilute brown agouti. But this shows the dilute mouse next to wild type black mouse. So it dilutes the coat color. It does not change the amount of melanin, though, in the skin. So again, how do we get to neurobiology? Another reason, in addition to this neural crest origin, is that Searle, way back in 1952, found a lethal allele of this dilute at this dilute locus that dies at 20 days after birth. And it has what are called, what look like classic tonic clonic epileptic seizures, with the most key feature is this epistotonus, which is this convulsive arching of the head and back. They have these starting at about 12 days and they die. So you'd say, well, this is why they die, but we really don't know that. Also, the Russells, a husband and wife team at Oak Ridge National Laboratory. So those of you from NCBS, we have something that's very parallel in the US. So the Atomic Energy Commission funds biology in NCBS. Well, the Department of Energy in the US funded this Oak Ridge National Laboratory that became a hotbed of mouse genetics. So they had a tester strain that was dilute and several other markers, and they did radiation mutagenesis experiments. So in the, by the 80s, this was actually the best understood region of a mammalian genome because of all these deletion mutants that could then be nested. And we had a region of 8 or 10 genes that was definitely the best understood. But the important point here is that these radiation-induced alleles, these were all the lethal. So we're pretty confident that that means that this is the no mutant phenotype, that no function gives you the seizure phenotype. And again, these melanocytes are derived from the neural crest, so that suggests a nice connection between coat color and the neurological phenotype. One thing that Peter didn't touch upon, and that is that we all have these little, I hope we all do, maybe I'm wrong, but we all have these little experiments in our closets that we did and we can't figure out how to package them for publication, or we don't think anyone will believe them. And I have a really important one with respect to the seizure phenotype. So when I first moved to UT Southwestern, I was next to the sleep lab, and they did, horrible things to cats with the electrodes and everything. And I talked these two guys into doing the same thing with my mutant mice, which were 10 days old, and probably a lot of you know about doing stereotactic electrode implantation in a rat. Well, this does not work in a 10-day-old mouse because the brain is like a potato chip. The skull is like a potato chip. There's no way to do it stereotactically, but we were pretty confident. We had one in the motor cortex, we had one in the cerebellum, and we had a control electrode. we saw absolutely no epileptiform activity. So what I know and have never published is this is not a seizure, this is not epilepsy, even though you can show a movie to a pediatric neurologist and they'll say, yep, that's a tonic-clonic seizure, no problem. So we don't really know what this neurological phenotype is.
Can you do EMG kind of thing?
An EMG, yeah, we did have one in the neck muscles. We couldn't see anything there abnormal other than, of course, movement artifacts, but no epileptiform activity at all. So it's possible that this could be peripheral. So again, my postdoc advisor showed that this co-color allele was caused by retroviral insertion, this classical one. So I was basically the last postdoc left standing when we finished cloning it, which was a huge task way back then. Its importance went up dramatically when a French group showed that Griscelli disease, which has the same symptoms in humans, it maps to the right region, and it was associated with mutations in the homologue in humans. So they have the same phenotype. They have diluted hair color, they have seizures, and they die very young. So this really established the medical relevance of this. But biologically, I'm not so sure. So again, we have pretty identical phenotype, which is nice. But you want to say the lethality means that it's really important. Would you agree with that? Okay. Here's a problem, though. It's not very lethal. The mice die at 20 days of age. When I was a postdoc and I did in situ hybridizations on embryos, there's huge amounts of this stuff being expressed in the entire brain and neural tube of a 9-day embryo. And there's no phenotype if it's gone until 3 weeks later. So the lethality, I think in some senses, has been confusing for a very long time. So again, 20 years later, it's really not clear what it does. And even worse, it's not clear how many different things it does. Again, here's another example. It's not axonal transport, but it's another example of how we get blinded to things. So if back in the 50s, when they looked at melanocytes from these mutants, so they did unstained section. That's the wonderful thing about working with melanocytes. You can do bright field microscopy. They did sections of skin and Harderian gland, and they saw that the wild-type melanocytes were very dendritic, and they saw that the mutant melanocytes were these big little spots of melanin, just very, very round spots. So the conclusion was that it was adendritic. And one of the authors here literally wrote the book called The Co-Colors of Mice. You can find that online where they describe all of these mutations, and it's kind of going back to a really interesting time in mouse fancy. So we actually had a decent idea back in the mid-90s, taking what we thought was true from the 50s, and said, well, do we really know this from sections? What if we just take these melanocytes out and culture them? And it turned out that if you look at the wild-type melanocytes, again, this is all just bright field, you see some dendrites. I don't know if you can see them, but they're dendritic. So that holds up in culture. When you do the mutant melanocytes, you see the clumping of the pigment. But if you do phase contrast, you still see that they have plenty of dendrites. So this was a huge difference in understanding what this does. We went from being convinced that it was involved in cell shape. I had all these little cartoons drawn that were all completely wrong, thinking that it was helping the power actin out into dendrites and helping to form the dendrite. Completely wrong. All it was a defect in organelle transport. So as Peter told you, what it appears to do is it appears to pin these melanosomes or retain them in the cortical actin, and when it's not present to grab them, dynein basically wins this tug of war, and you get the clumping around the nucleus. So again, why are some proteins better than others? In this case, if you look back through the history, it's because we had a mutant that had a phenotype, and we had a human phenotype that corresponded, and we had a tag that allowed us to clone it really early, so a lot of people could work on this. Now, finally, after all that, let's get to axonal transport. So this was a really landmark paper in 1992. I'll show you the actual movies from it. This is the one I had suggested that you read. How many have had time to look at it? Okay, that's fine. We can go through it. But what they found basically is that so Scott has introduced the extruded axoplasm to you, correct, as a system? What they found was they found actin networks if they looked with rhodamine phalloidin, and over time these actin networks would spread out from the edge of the microtubules, and they would become thicker over time. So this is a kind of artificial preparation, but they see this actin spreading out, confirmed by phalloidin. And you can see microtubules in it that aren't stained by phalloidin, for example. Here's one movie. This just shows their axoplasm itself. There's my person. Just to show the motility they see, and you can see the microtubules by DIC here, and things moving very rapidly, all sorts of different sizes. Then this shows this. I'll actually show you this. The static images are actually more helpful from this than this, but they watched things moving along microtubules, which you can see by DIC, and they also took tracks off the microtubules on invisible tracks that they couldn't detect. And finally, this shows things moving on their preparation of actin. So they had things moving along actin. They're pretty sure it's actin. But here's a couple of examples of things actually moving on microtubules and then popping off. So here you can see a microtubule by DIC. Here's an organelle moving along it. 1, 2, 3, 4, 5. And here it moves off it, and you can see there's no underlying microtubule. So the idea is that things are, again, this goes back to what Peter was talking about, that we thought that the myosin, especially the myosin 5, is some sort of ancillary thing that if you think about, I would say FedEx here, but no one likes FedEx. Let's say DHL or Blue Dot. You have a plane, which would represent the kinesins and dyneins that would deliver your package. It's all addressed neat and tidy. It delivers your package, let's say, to the presynaptic region or the growth cone. And from there, it gets dropped off to the local delivery truck, which is myosin 5, which then reads the same address somehow and actually does directed movement and gets it to where it needs to go. And I think as we're maturing, our view of that is now that it's a much more sloppy process. But no one attacked me for that. So here we have actin-dependent movement. The first motor you would look for then would be the myosin 5. And George Langford's lab found evidence that they could inhibit it, for example, with anti-myosin 5 antibodies. Scott and his people were co-authors on a paper with my former advisors where they showed a direct interaction with a kinesin by 2 hybrid and cellular assays. And Peter mentioned the work from his lab showing that it's involved in mitochondrial transport. Probably some of those are mitochondrial. But I want to make a point here to go back to that toolbox size issue because, for example, a student in Jim Sputich's lab read these papers and said, oh, you know, Jim knows John. He goes to Montana. John has all these mutant mice. I'll look in the mutant mice for defects in mitochondrial transport. Well, she came and spent 3 weeks in my lab, made beautiful neuronal cultures from the null mutant and the wild-type control mice, and saw absolutely nothing. Just absolutely nothing. It's one of those rare cases where a thesis project gets killed in 3 weeks.
You saw nothing. What do you mean by that?
Absolutely no difference. Yeah, no difference in motility of mitochondria in the mutant mice. Now, I'm not saying that Peter's wrong. Again, we have different toolkits, and it can compensate probably because we have so many more myosins. So again, so here's a movie from George Langford from around, I believe it's 2003. All the faculty have seen this. I forget exactly when he did this, but here you have myosin 5 and kinesin attaching to the same vesicle. And again, this is just a cartoon illustrating this kind of directed transport addressing idea. And so for some reason, the myosin 5 is being dragged along behind like a sled. But here, then things switch, boom, boom, nice and clean, and it switches over to the actin filament, and these little white flashes are ATP hydrolysis. So while it seems kind of quaint to us now, this made perfect sense to us at the time. I mean, I think everyone would agree with it, that this is how we were thinking of things. So that gives you an idea about myosin 5, but what I'd like to say, what I want to keep coming back to is that I don't think what Peter said is crazy. I think what myosin 5 does in axonal transport is much more like what it does in the melanocyte. That things encounter actin, you have vesicles with myosin on them, they encounter actin, they go onto the actin sometimes. And the frequency with which they come on and go off depends on the relative amounts of microtubules versus actin filaments, and the relative amounts of myosin versus microtubule-based motors on the vesicles. So it's, I think, while that's sloppier and it doesn't appeal to us as much as engineers, it probably is more like reality, that that would be the way that evolution would overcome roadblocks and things, that things would occasionally just go off and have another place to go for a while. Yes?
Do we have direct evidence of this happening, the switching of kinase myosin?
The most direct evidence would be that 92 paper. And then in subsequent papers, they could show that that activity appeared to be mediated by myosin 5. That's pretty good. They have pretty good evidence for that. But that's, yes?
Wasn't there some subsequent criticism about that imaging that George did that about switching from microtubules to actin?
Tell me what you know about it, because I was a mouse geneticist then. So.
Yeah, well, there are a couple of issues. Yeah, there are a couple of issues. I mentioned the issue about time lapse. You've also brought that topic up. What kinds of outcomes do you see? One of the things that you'll notice in the ones where we see the movements gliding along the component is that it contrasts very dramatically from the earlier images where you see all the grounding.
Mm-hmm.
So what they've done is that by doing time lapse, it's done essentially a high frequency filter. So it cancels out all of the high frequency movements, just like you were saying with the And so what happens is that you have to be careful, because if you look at the vesicles that happen to stay close to the surface, because you're only looking at one plane, if you see that they're moving close to the surface, it looks like they're gliding. And if you're not seeing what they're gliding on, well, it's a perfectly reasonable interpretation. So they must be gliding with something too small to be seen.
Mm-hmm.
And they did show that there was actin, so that much is true. No question about that. But the problem, though, is that the actin was meshed with it.
Right.
And they were looking like they were moving nice and smoothly at a fairly long maturity.
That shouldn't have happened on this one.
You would not expect. So it's problematic, and no one's ever been able, no one's ever gone back and documented it. And it goes back to the importance of understanding the strengths and weaknesses of the method. And that's actually a topic that is well worth discussing.
But in the system where they only had the actin filaments, that wouldn't apply, right? Because so it can be, the switching is the only.
But you still have the problem, because what happens is that it happens to be confined, it happens to stay close, and there's just a probability of routing motion that some particles will remain close to the coast. But they will stay there, and they will essentially be moving like in this apparent reliable motion. Now, what they need to do is they need to do something like inhibit enough causing this be stuck to those. They need to have some other method for showing that there's a direct interaction for that. They need to show that it goes away when you have the addition of So there are various things that one can do with this, and it's, but you just have to, you can't just use the gliding per se. If you're doing time lapse, and that's the problem.
Doesn't Gelfand has a few papers kind of showing that if you remove all the tubulant from his model system, you still have, like the jiggity movement of vesicles, melanocytes.
Well, and John Hammer showed with mammalian melanocytes that what wasn't happening in the mutant, but he showed these peripheral things where the melanosomes appeared to be wandering around aimlessly, which is what I kind of like that idea, because I think the movement is almost an artifact in terms of the function of myosin 5 in both of these systems.
Just to keep them evenly distributed.
Right. It's just
wandering around on an actin mesh is the perfect way to do that.
Right.
One puzzling thing for me has been that if you look along the axon in cultured neurons, there's not that much actin in there, right? I mean, you stain it with collagen or whatever, there's a little bit there. I mean, most of there's microtubules in there. And it looks like, of course, the actin is like a donut-shaped thing.
Right.
Go up and down, kind of see it.
There is a meshwork of actin though surrounding those.
Yeah, the cortex.
Right. But it's so less, right?
Well, it's
Just
not
like
as dense as the plasma membrane size still, but it's there. And it's actually in the axoplasm, it's been directly measured by Morrison Lassen.
Yeah.
And looking at the levels there, and it turns out that it's a fairly significant amount of actin. And it's also true that if you take this axoplasm and disperse it, what you find, or actually if you don't have to disperse it, you can just put it in a gel solvent, which cleaves like your filaments, and do that in the axoplasm. Is that the vesicles will continue moving along the microtubules, but when they fall off, you can't find them. So you start, all the vesicles start drifting away. So yeah, I think that's a good question that actin plays an important role in organization. I suspect that obviously the myosins can play a role in certain things like the membrane approach, the details they don't see. But the big problem with actin is that it's just not organized in a way that will allow you to make long excursions. Anything but very few cell types, most of those.
Right, and so even though myosins, it's been shown by many, many labs with beautiful, tight experiments that myosin can take long excursions on an actin filament. Why did evolution select for that? Did it select for it so that it could take long excursions on actin filaments, or is it some sort of adhesive, traffic jam ameliorating property?
It may be just to increase the efficiency of the motor, even though it's dealing with these short actin filaments.
And to disperse the motors too. Yeah.
That's true. It's good to think out of the box, though, because just because someone has proposed something that's sort of the great assumption that you probably do, you can be led down the dark path so quickly if you try to go out and find experiments that support what you want to do. What you should always be doing is challenging quality, finding things that will essentially be a way to disprove your hypothesis, not trying to prove it. Because if you're trying to prove it, you're going to get yourself in trouble.
Right. Yes.
In the movie, probably they would have analyzed the data slightly differently because if you have calculated the mean square displacement of each particle as a function of time, then you can tell, you basically whether it's diffusion or whether there is some active component is found. So if there is an active component to it, you can get, for example, super diffusivity, even if actin is measured.
You would probably want to do much finer time sampling.
Yeah.
Before you
That's a good goal.
And then
high frequency movements.
Okay, so I'm going to kind of ignore most of the other myosins. Most of them are present in the nervous system, but there's not really great evidence that any of them are performing very nervous system specific roles. So you have myosin 1s that are attaching to vesicles and controlling actin reorganization. All these things are there, but they seem to be functioning more in the same sort of context as they do in fibroblasts. That's probably wrong. I'll add. So we have this putative function for myosin 5. It's very solid. Many people studied it in the melanocytes. As you can tell, it was just revealed to me that there is some skepticism about the data in axoplasm, but as me, for someone from outside the field, you just want to gobble that up and say, hey, my thing is more important now. And this shows the danger of that. But then all these other things came up for myosin 5. It's a major component of the postsynaptic density, and that was found in a landmark mass spec paper from Mary Kennedy's lab. And let me make a point about reagents too, because I'm not sure it's been made, but the best myosin 5A antibody in existence comes from this study, because Russell Laliconis wanted to try to make monoclonal antibodies, so he had immunized some mice. The fusions didn't work, but we actually have these tiny amounts of mouse polyclonal antibody from the bleeds that he did. And in our experience, almost all of the commercial antibodies are terrible for myosin 5A, and this is the best one, and we have microliters of it, and we're not going to share it. So these are bleeds of a mouse. Think about it. Just tiny things, and it works so much better than anything else we've found. There are many studies on secretion in chromaffin cells, which is easier to study than synaptic vesicle secretion. Tony Brown, whom you've heard mentioned, it showed that it's involved in neurofilament transport. One paper I'd like to talk about is a fairly recent paper from Don Arnold's lab, showing that it seems to be involved in targeting of membrane proteins to dendrites, and also in transporting MRNPs, ribonucleoproteins. And there's really nice, again, this gets back to what Scott said about seeing the things you want to see, but this is really attractive because there's a huge literature on Myo2P, which is a member of the same family in yeast, and it transports mRNAs to the bud site. This is true point-to-point transport. However, yeast actin is organized in cables. This is a really important point. So the actin is arranged in a way where you can get that point-to-point transport. I just learned a couple of weeks ago, Benny Sheila came and gave a talk. He's found epithelial actin cables, and it appears that a myosin 5 is doing directed transport in that case. So I had started preparing this talk, and there's already been an exception to it. The other two things I'll talk about, and these were the papers that were assigned, as I understand. It's myosin 5's involvement in long-term potentiation, and there's a big disagreement here. We're co-authors on this paper showing that myosin 5B is involved. Then a paper came out shortly thereafter concluding that myosin A is responsible for this. But first, before I get to that, so that's an outline of this portion of the talk. First, let's go to this paper here, the targeting of membrane proteins. Don Arnold's lab found that overexpressing the myosin 5A tail prevents dendritic accumulation of these dendritic proteins, the endogenous ones. So if you overexpress the tail, you look at the localization, you look at dendritic versus total or dendritic versus axonal distribution, it's much more homogeneous than it was. Then to do a positive test of this, they fused CD8 as a marker to melanophilin, which they know interacts with the myosin 5A tail, and sure enough, that caused the CD8 to accumulate in the membranes of dendrites. So this is a fairly weak experiment. This is a pretty strong experiment. And again, so it looks like this could be trapping. So the myosin 5A could be trapping it in dendrites. And so I'm going to try to weave all these together in a very awkward story, but to illustrate, what I'm trying to illustrate is the weaknesses of genetics and the weaknesses of these phenotypes, because these really complex systems have ways to compensate. So of course, I saw this paper. I probably have 90% of the myosin 5A null-mutant mice, the lethal mice in the world, so I immediately called Don Arnold to propose looking at, that he would look at neurons from our mice. And he said, we tried that. It didn't work. Anyone know why? Ideas? Come on. Why wouldn't it work?
That's kind of vague. What do you mean it didn't work?
What, can you offer some biological reasons why the mutant experiment did not work?
What was the experiment?
That they looked in neurons from mutant mice, and these proteins were perfectly fine. They were still localized to dendrites. Sorry about that.
The developmental age at which these proteins are normally expressed?
These were the endogenous proteins. At least he said this is a phone conversation. That
so these proteins are expressed from like day one in cultured neurons?
I'm not sure. Yeah, that's one possibility. Anyone else?
So is there any other isoform of the myosin which is actually taking care of the function of the...
Yeah, for example, there could be myosin 5B there that somehow does it. Maybe the proteins that interact with the tail, it's possible that there's...
Where is the mutation in this myosin 5B?
It's null. There's no poly A messenger RNA produced. It's a deletion, a small deletion in the tail, but if you look at the RNA level, there's basically nothing.
Is there compensation with the other myosins?
Yeah, you could upregulate the other myosins. That was the thing. Remember I told you about Jim Sputich's student who had the 3-week failure? She was just clawing, just grasping at straws, saying, well, maybe it's compensation. Maybe we should look at myosin 5B. And we all kind of tried to talk her out of it and say, drop that project. Because it could be, but it's going to be very hard to show. But I'm going to come back to this because he told me another story about the review of the paper. They have a paper in 2011 where they did the converse, showing that myosin 6 is involved in axonal targeting of membrane proteins. So I think it's been presented to you that the myosin 6 is the oddball group that's minus N directed. Goes the opposite way, it's involved in endocytosis. They looked at a larger sample of axonal proteins, and they expressed a dominant negative myosin 6, and they reduced the targeting. If they fuse markers to myosin 6 binding proteins, I mean, it's almost exactly the same paper with the players changed. That promoted axonal targeting of proteins that wouldn't normally go to axons. And so the mechanism here is that the axonal proteins that are carried to the dendrites tend to get unloaded and then subsequently reloaded by endocytosis, and then those vesicles get transported to the axon. So they think that this is working by dendritic endocytosis. So here's 2 of them that seem to be involved in sorting membrane proteins. So, let's see. So again, the MRNPs, the Myo2P makes it attractive. What I'm going to talk about up at the Hill Station that I can't pronounce is that we have really, I think, solid data. I'll be interested to see what Scott, especially Scott, thinks. I'm sure he'll tell me exactly what he thinks about it. Showing that myosin 5A is required for transferring RNA from Schwann cells to the axons they ensheath. So it's kind of a solution, partial solution for this problem of axonal transport. How do you supply everything to the end of the axon? We think that there might be a supplement, not a replacement. I've offended several people by saying this. We're not saying that this replaces axonal transport, but it's a supplement to it by getting extra things into the axon through a very short path that involves transfer of an organelle from one cell to another. And if the other thing that makes that attractive is that we've already implicated myosin 5A in cell-to-cell organelle transfer because it's required for keeping things at the cortex of the melanocyte, and from there melanosomes are transferred to other cells. So the whole thing lines up very nicely. Now I'll go on to talk about the papers on long-term potentiation, and I guess we're having a session where you'll read these and we'll talk about them. And what I would like you to do, I don't really expect anyone to get the answer. I'd like you, this is an example of trying to accept things that are true. If you accept both of these papers, they can't both be correct. So we can talk individually or as a group about how we can reconcile these data. One thing is that we used a much tighter method to study myosin 5B, so it's a much more positive method, whereas, oops, I'll get to that later. I'll get to that later. So here's the background for looking at myosin 5B in long-term potentiation. Everyone understands long-term potentiation, the basics of it? Anyone doesn't? So, this is basically understood to be the basis of learning and memory. If you do high-frequency stimulation of a synapse, you get a strengthening of that synapse. And there are several mechanisms. I think, as an outsider, again, my understanding of LTP is that it's so important that evolution has grabbed every possible mechanism to participate in it. And you'll find so many papers saying, oh, this shuts down LTP, this does, it's a major player. There are just so many major players, and I think that's an evolutionary function. But one of the major aspects of long-term potentiation is that upon the stimulation, endocytic recycling puts more GLUR1, glutamate receptor subunit, into the plasma membrane. Okay, that by itself strengthens that synapse. And what Mike Ehlers found was that these endosomes have Rab11, and we know that Rab11 is a very strong binding partner for the tail of myosin 5B. So Mike contacted me because we had been working on myosin 5B as well. And I actually like Mike's drawing of what we did. I'm assuming it's Mike and not someone in his lab. But there's a whole series of papers, but what we did, we were really unsatisfied with this idea of overexpressing a tail of a motor. Because what that does, think about it mechanistically. What does, if we overexpress the tail of the motor, what does that do to the cargo track relationship? What are we doing? Anyone?
So it can bind to the cargo vesicles and probably can quench it from binding to them.
Right, so what does it do to the real cargo in the real tracks?
So it will prevent it from, it will take it actually away from the track.
Right, it disconnects it. Okay. So that's dominant negative, right? Is that the only way you can get a dominant negative effect?
It can be reversed also, expressing just the motor domain. No, but that will, yeah, but that can affect the myosin 5 motor.
Yes, it could, but there are lots of myosins. Yeah. It's, what about, any of you familiar with what happens to your muscles after death? Obviously, it hasn't happened to any of you. Anyone? Pardon me? Rigor. Okay, and that's caused by? Lack of ATP. What does ATP do to a mixture of myosin and actin? Detaches, okay? You use that in purification all the time. You attach it, you pellet it, then you detach it, and you get the live stuff off. Okay, so what is bound to the myosin during rigor mortis, probably? Pardon me? Okay, any others? Won't it go through the cycle? Okay, so ADP is bound, right? So if you have ADP and myosin, it's going to lock on. So we, in collaboration with Peter Gillespie, who was a colleague of mine at UT Southwestern, and Kayvon Shokat, who did this for kinases. It was very convenient because we just basically took all the chemistry they did for kinases. But it turns out, and this never ceases to amaze me, that you can, so here's your nucleotide, here's the myosin head. it's blue, you can mutate a tyrosine in the ATP binding pocket that's in very, very close proximity, if not bonding to the N6 of the nucleotide. So you're in the active site of an enzyme. This is a really important residue. It's almost universally conserved. You change that tyrosine to glycine, which is the biggest size change you can make without changing charge, right? A whopping change in the configuration of this ATP binding pocket. So that's illustrated by this little extra sliver of space here. And here's what's amazing, is this thing still works. So you can purify it, you can test it in vitro. Really the only thing it does is when you purify it and put it in the fridge, it's about 10-fold less stable. So it goes bad more quickly. But with another myosin that we did this with, myosin 1C, we've actually made a knock-in mouse, a homozygous knock-in mouse that's perfectly normal. So just to give you an idea of how these things come about, though, Peter and I knew each other at UT Southwestern. Then he went and started his own lab. And he called me because he wanted to study myosin 1C and hair cell adaptation. This is when you actually adjust the opening and closing of the channels. That part's not important. But we talked a lot, and we came up with two methods to do this. One was by turning off transgenes with the myosin 1C using tetracycline control, TET-on, TET-off technologies, which there are a lot of papers using them, but in the mouse genetics grapevine, you also hear of a lot of cases when they don't work. And that's when you got to listen to people with this publication bias, because people don't publish their failures. On the other hand, Peter was talking to Kayvon Shokat. They had been undergrads together, and Kayvon had this system for doing the same thing with kinases, and Peter told me about that. And I have to be really honest here and tell you that the TET-on, TET-off thing for the mouse guy, that was a lot of work for me. This chemical genetic thing where you had to test a bunch of analogs in ATPase assays, that was a lot of work for Peter, the biochemist. So my arguments in favor of doing this were entirely selfish, right? Because this was a lot less work for me. And I talked Peter into going this route, and it worked beautifully with myosin 1C. We did it with myosin 5B. So you have this extra space. It still works. The mutant still works.
So will it work in fly also if you make a mutant?
I would. So I can tell you our track record is myosin 1C worked beautifully. Myosin 5B did not work. It did work beautifully, even better. And myosin 5A did not work.
Nice.
So we're 2 for 3.
And what's the major difference in the motor domain structure?
None that we can identify. It has some differences, but I've talked to crystallographers, and they say that the residues that are different are on the outside, and they don't care about them.
So I think speaking of some...
Okay, I'll stand back here. So Kayvon Shokat's lab then supplied us with N6 subs. Remember this tyrosine is there with N6. He provided us with N6 substituted analogs with all sorts of different doodads on the N6 group. And you don't know the structure well enough to know which one will work. But for example, for myosin 1C, Peter tested 21. For myosin 5B, we tested 5 and got one that worked beautifully. So in the presence of this ADP analog, it can't get into the wild type binding pocket. But it can get into the mutant. And so we've tested it. We know that in ATPase assays, we know that it can inhibit ATPase in the mutant and has no effect on the wild type. So what will you do to the heads in that case? Anybody?
In wild type or mutant?
In the mutant.
It'll lock on to the
It'll lock on. So we'll lock on to the active filament. Is that a dominant negative phenotype? Yes. Is it different from overexpressing a tail? Completely, right? But they're both dominant negative. So here we're stopping everything, but it's completely specific because this will not fit into any other myosins that we know of because we can inject this into our control cells as well.
It's also better because you understand the mechanism, right? Dominant negative, you throw it in, you have no idea what's
Right. it could bind to completely irrelevant things too.
You don't really know whether it's binding or doing something in a cell.
And for example, I can talk to you, if you're interested, I'll talk to you about it later, but we use this method, again, overexpressing a tail suggested that this myosin 5B was involved in getting transferrin in the classical transferrin recycling pathway from perinuclear compartments to the plasma membrane, and we found that that was all wrong. But it wasn't until we had the data that we thought about it a different way. So anyway, we're stopping it. This is a real dominant negative. And here are the LTP data. You do the high frequency stimulation. You have a baseline here. And what's really nice about this is we've made transgenic mice expressing this after doing all this in cells with transferrin. We have we call it wild type, but we've actually expressed the wild type in a transgenic mouse just to raise the level a bit because you can get weird effects just from overexpression. So it's dialyzed in then to the postsynaptic neuron. So if we have the wild type, we get very good LTP. This just shows the currents you get after the increase in currents you get after the high frequency stimulation. If you have the transgenic without the analog, this shows that mutation is doing nothing to LTP. It's the same as wild type. And however, if you have the transgenic mouse expressing the sensitized mutant protein with the analog, we really knock it down.
So in this case, you just have the analog in the recording method?
Right, you're dialyzing it in the recording pipette. And in the case of hair cells, we had this beautiful temporal control because it took so long for it to diffuse from the cell body to the tips of the stereocilia. So we could be sure that the cell we were recording from was good, was healthy. So here we get to an interesting point. Here's Mike Ehlers. He's very, very good, an authority in LTP and recycling. He's now working for, not Novartis, it's the other big pharma. Pardon me? Merck, yeah. And this has been shown by several labs with highly purified myosins. If you look at them, you can see, you can detect interactions between the head and the tail. It's very solid in vitro, but no one's ever shown this happening in vivo. A similar thing has been shown for non-muscle myosin 2 over 20 years ago. No one has since shown that it has any effect in vivo. But let's think about this, because this is, you have the whole thing folding up and locking up. How is this drawn here? You see you have one head attached. What would it likely do?
I thought it will dissociate from the actin.
Right, it would be soluble then, right?
Remain in the soluble.
Okay. So in low calcium, this is shown attached, but it probably wouldn't be attached. It would be soluble. And in high calcium, that induces it to unfold, and then you get actual transport of GluR1 subunits to the plasma membrane, again, facilitating LTP. So the big problem is this folding. It's never been shown in vivo. Here's another diagram of it. Again, showing the folding in response to calcium or response to cargo binding. Both have been shown in vitro. And there was even an experiment in the paper where they shortened it so that the tails couldn't reach the heads. So this wouldn't be able to fold up. And it gave the expected results. But again, if you're looking for the expected results, that might be dangerous. I'll show anyone what happens if you express this in a fibroblast, what it looks like. It's very, very interesting. So again, does this happen in vitro? What predictions? We have the solubility. Here's why I'm skeptical. Because we've expressed the same sensitized mutant with the GFP tag. Again, at sub-endogenous levels, we're not overexpressing. So what would you predict if you're folding up? Where should we see this GFP signal? Anyone? So it should be diffuse and soluble. Okay, now we're getting back to the point I want to make about myosin being more of a kind of a brake or a dynamic tether. This is an experiment Bill Province did in my lab. Again, this is the GFP signal you see. The cell on the left will be injected with the analog, the cell on the right will not. And of course, we've done all of the 3 controls as well. What? No. No. There we go.
I think it's picking up something on overlaid on top of that. You probably have to click on the movie, then.
There we go. Okay, this is before injection. You can see up here after injection, everything stops. It's not soluble, right? You're labeling all sorts of particulate, punctate things here. There's very little background for it.
Although it dipped a little bit. I think the puncta.
Well, we've diluted it somewhat because we've injected the analog, so we've changed the shape of the cell. I think that's a lot of it. It's a little bit bigger now. But the particles have stopped. So I had a group of summer students that I had measure the speed of all of these, and even things that were moving along microtubules that would be 1 micron per second or more, they all stopped. So the idea is that you're zipping along in a microtubule, and the actin clearly can encounter, the myosin can clearly encounter actin very quickly, lock on in a Riger state, and stop the microtubule-based motion. So it turns out, again, like with the mitochondria, if we treat with latrunculin and depolymerize actin, we increase net speeds.
So if you do that experiment and inject, what happens? If you remove the actin, inject the analog.
We haven't done that. We've injected the analog into cells expressing wild type, and it has no effect. And we've mock injected, of course.
That means there is a kind of tug of war going on between these kinesins and myosins at the stationary level these are binding to.
And right out in the middle of the cytoplasm, which should be fairly microtubule rich.
All along axon, not only at the synapse.
Right. That's essentially what I'm trying to get across. So that Peter Hollenbeck is not crazy.
It means all these modules are bind every time on the mitochondria. It's not that it's just going or coming. And whenever it comes across the actin or whenever the affinity increases, or you can say the affinity of kinesin is decreasing, then it binds to actin
and so the way I see it, and I'd like to hear a discussion about it, is I don't see that there has to be a really active switching mechanism. That you have different concentrations of actin versus microtubules in different parts of a cell, and that dictates where your organelles will end up.
Encounter rather than a complex switching.
So one of the issues is that what exactly, so this is GFP myosin 5B, right?
5B, yeah.
This is GFP myosin 5B. So each of these puncta that you're looking at are a bunch of myosin molecules.
Right.
So are they really, they're probably on a vesicle, right? So what
yes.
you're really looking at is a vesicle coated with myosin 5B.
Mm-hmm.
So in your left image where you're injecting your compound, I was wondering if you create a situation where you prevent the association of the motor with the actin filament, that could still explain your phenotype here, because what you might be getting are vesicles that still have the myosins attached to them.
Mm-hmm.
They are still away from the actin filament, just as your model predicted. But it's not so much that the stationary puncta means that they are not solubilizing. Maybe they are solubilizing, but they're still associated with.
Right. I don't like the folding model. I think they're unfolded all the time and available all the time. I'm offering this as evidence against it. Okay. So again, we'll talk about these long-term potentiation papers later. I should finish up. But so I'll tell you what, I told you what we did in this case. We have the myosin 5B. Let me go back just quickly, one thing. So my model for what's going on is much simpler. All we need in a dendritic spine is that the increase in calcium makes it more likely that the head lets go. And then diffusion will get more of it into the plasma membrane. So it's actually being sequestered from the plasma membrane, again, instead of point-to-point transport. So I'll leave the papers for you to read. I've shown you one problem with it, but I do still think that myosin 5B is the one. And I'll leave it to you to look at this myosin 5A paper, and I'll note that they did myosin 5B negative controls, while we did not do myosin 5A negative controls. And I think it'll be fun. I don't expect anyone to, the reason is very, very obscure. And again, you have to read the primary papers, and in this case, you have to go back several papers too. So, in closing, I'd like to say I don't believe Peter Hollenbeck is crazy. Because when you think about it, how many cases of point-to-point transport by myosins exist in higher eukaryotes? And I now have to amend that based on the talk I just heard from Benny Shilo. How many cases of point-to-point transport by myosins exist in higher eukaryotes that do not involve actin cables? And the answer is, it appears to be 0. So here's another paper. So again, he was not crazy. Here's a paper I'd really like you to read. Oh, I don't have the citation there. It's Trends in Cell Biology. Bill Bennett argues makes the same argument, that these myosins don't really move things around. They're very dynamic tethers, and if we think about all the things they do, as you heard, that these things walk. And so I'd like to point out that metaphor is a little dangerous. Because what else when you walk across a room, how do you do it? You walk straight across, right? Every step is directed. When a myosin walks along an actin filament, how does it walk? We have lots of processivity, right? But what happens occasionally? Have you looked at any of these myosin papers? So what happens is occasionally it takes a step back. You never do that when you walk. So that's a defect. So the metaphors always break down. That's the danger. The cartoons break down, the metaphors break down as well. And if you have a myosin that does this, that can walk forward, that can walk backward, that walks backward very well if you're pulling on it, but it also, some of these myosins can also kind of shut down, and the simplest way to think of it is that, let's say the nucleotide binding pocket is in my back, and as I'm stretched backward, that closes the nucleotide binding pocket so it can stay bound to ADP.
Actually, Yanagida has this raging debate with
oh yeah, it's fun, yeah. But this is true for myosin 1C as well, and we think that's how it's working in the adaptation. But the idea that you can pull it and it has this strain responsiveness, it's an ideal thing for this kind of dynamic tether where you're zooming along on a microtubule. Oh, I'll stop here for a bit. The microtubule can, the kinesin or the dynein can pull on you, and you'll take steps back on the actin. You'll break loose. It's a way to break up traffic jams without designing anything. It's the way evolution works. Yeah.
So I mean, if you know that kinesin, dynein, all of them move backward and the
Yes.
How different it is for unconventional myosins?
I think the big difference is that you don't have the polarity. I think all the same things operate with the microtubule-based motors, but you don't have the polarity with the myosin. And I mean, I work on these things, and it's kind of sad to say that I think they're kind of degenerate in that way. That you have this beautiful thing, you have a yeast that moves things from point A to point B, and it's like kinesins took over that function in higher eukaryotes.
How will you be able to differentiate between stepping back of the motor and bidirectional? I mean, you said that it's not...
Oh, because the stepping back occurs less frequently. Again, it's a probability thing. The stepping back occurs less frequently, but if you apply force, you can actually get a backwards walk.
Will there be flipping of the...
No, not that no.
Because myosin 6, I remember, I think it can move bidirectionally.
Yes. They all can move bidirectionally. It's where the preponderance of movement is.
Do they generate force in both directions?
No, I would say not.
Yeah, that's what Dietmar Malmsteen showed, actually, by flipping the neck linker.
Right, and that explained the myosin 6 reversal. So I'll leave you with that. But I hope you get a flavor for how you can maneuver and drop into a completely new field. And I'd say in a lot of ways I misunderstood things, as you heard from the discussion about the Langford paper. And I think in a lot of ways it gives me an advantage being from the outside. And I'll stop there. Thanks.
So, yeah, wait. Students stay back for a while. Faculties can have their lunch.
I think I made a mistake in one of the references. I hope the people are doing the
Great.
right.
It's really, I never thought it in this way. So it's really nice.
There, it's off.
And for all of you who had papers here, who've been assigned papers which have come at various faculty, please make sure that you discuss it with the most appropriate person before you present. Remember, 15-minute time.
Something interesting occurred to me when you talked about the Don Arnold myosin 6. The actin filaments in the axon are so dinky, no one's really been able to talk about polarity.
Yeah.
But 100 years ago, when Bob Morrison knocked out all the microtubules and established axons in culture, we did get movement. It was slow, it was not persistent, and it was net retrograde. And we assumed it was myosin 5, and so we predicted that the polarity was something like 3-to-1, plus ends of actin toward the cell body versus out. And that would help to explain Don's data.
Yeah.
Because that myosin 6 then ought to take stuff into the axon. If the movements that we saw 3 times as much movement, time of movement back toward the cell body when we eliminated microtubules from axon as outward. And so we made a guess in the discussion in that paper.
Yeah.
It's mixed polarity because they're moving both directions because we didn't know about minus-end-directed myosins then. And we bet it's 3-to-1.
Yeah. It's all.
In the group, 3 people are going to be there to field questions. All 3 people are looking for
Yes.